This presentation introduces a novel microscopy technique for resolving the spatial organization and dynamic behavior of proteins at the surface of living cells. Traditional resolution of fluorescence microscopy is limited by the diffraction of light. When molecules are closer than about 200 nanometers, they are seen not resolved due to the overlap of their diffraction patterns. This makes it nearly impossible to study how proteins are organized on the cellular membrane. Newly emerged super-resolution techniques like DStorm and STED have pushed resolution into the tens of nanometers. These methods rely on selective excitation of individual fluorophores and mathematical reconstruction of the resulting image. The idea behind these methods is simple. Excite fluorophores, preferably one at a time. Capture each image and reconstruct a high-resolution composite image. Despite the achieved resolution, these systems carry significant limitations. First, they're technically demanding, which drives up both complexity and cost. More critically, they're restricted to static specimens, meaning live cell imaging simply isn't possible. And finally, the intense light quenching used to reduce the number of excited fluorophores leads to rapid photobleaching. Moreover, the use of the above-described super-resolution methods does not guarantee unambiguous determination of the spatial proximity of proteins even in fixed cells, since the cell membrane is curved and contains many elongated protrusions, which complicates interpretation of data. The quantum nature of light opens new horizons in microscopy. Instead of measuring light intensity, we focus on detecting individual photons. Using time-correlated single-photon counting, each photon is tagged with two timestamps, one marking the time from the start of the experiment and the other from the excitation pulse. This allows us to unambiguously assign emitted photons to their excitation cycle for each emission channel and to correlate them. This simulation shows cross-correlation measurements in action. The red ellipsoid represents the confocal volume. A minor fraction of the protein population is tagged with red and green fluorescent probes, ensuring that, on average, only one or no tagged molecule occupies the confocal volume at any given time. Yellow dots indicate complexes formed by the proteins tagged with red and green probes. This color scheme is applied to the autocorrelation and cross-correlation functions shown in the lower panel. When two labeled proteins enter the volume together, their emitted photons become cross-correlated. The yellow cross-correlation curve displays this cross-correlation. The stronger the coordination between the labeled proteins, the greater the amplitude of the cross-correlation function. The red and green curves show the autocorrelation functions for each labeled protein. The shape of these functions allows us to determine the dynamics of protein diffusion within the membrane. The microscope uses a 405 nanometers picosecond laser and a dichroic beam splitter to separate excitation and emission light. Emitted photons are split into two channels, each tuned to detect a different quantum dot. Using quantum dots solves two key challenges. They enable single-beam excitation, making the instrument relatively easy to set up and operate, and they offer high brightness, which is needed for single-molecule detection. With this method, we're not analyzing pixel intensity. We're counting photons and determining their timing. The raw data from the free quantum dot experiment are shown in the top panel. The bottom panel shows the photon counts corresponding to bound quantum dots. The cross-correlation functions presented in sections B show high amplitude for bound quantum dots and near zero amplitude for free quantum dots. The fluorescence decay curves in sections A indicate that the fluorescence lifetime of these quantum dots is in the tens of nanoseconds range. Although the principle looks simple, there are technical challenges to consider. Fluorescence probes emit in all directions. but only a fraction of this radiation is collected. Detectors have limited efficiency and generate dark counts. To reliably detect a fluorophore, it must remain within the confocal volume for at least 10 excitation cycles, or 1 microsecond at a 10 MHz laser rate. In addition, the data are affected by background fluorescence. Fortunately, we can clean the data using photon statistics. Quantum dot fluorescence lifetimes are in the tens of nanoseconds range, much longer than background emission, which last only a few nanoseconds. Dark counts have distinct time density profiles. By applying sequential, the lifetime and dark counts filtering, we separate true signal from noise, ensuring that the counts in the histogram reflect quantum dot emission only. T-cell functional avidity maturation was a long-standing problem in immunology. T-cells recognize and eliminate infected cells. Each clone expresses a specific type of T-cell receptor tailored to its target antigen, while all T-cells share an invariant CD8 co-receptor. A long-standing observation showed that antigen-experienced T-cells exhibit significantly enhanced cytotoxicity compared to naive cells, yet the underlying mechanism remained unclear. We hypothesized that this is due to a reorganization of TCR and CD8 on the cell surface. This hypothesis was confirmed for the first time through the development of this super-resolution microscopy technique. This schematic shows how TCR and CD8 interact with the MHC antigen on an antigen-presenting cell. To test our hypothesis, we labeled TCR with 800 nanometer quantum dots and CD8 with 665 nanometer dots. These fluorescence probes have minimum crosstalk and can be excited by the same wavelength, allowing the cross-correlation measurements. In naive T cells, TCR and CD8 are distributed randomly across the membrane. Upon antigen exposure, however, their localization becomes highly coordinated, reflecting a spatial reorganization of these receptors in antigen-experienced cells. Quantitative analysis of peak amplitudes indicates that each TCR is typically surrounded by 2 to 3 CD8 molecules, consistent with a 1 to 2 or 1 to 3 stoichiometry. CD8 binds MHC non-specifically and at a significantly faster rate than TCR. When in close proximity, CD8 promotes subsequent TCR engagement and stabilizes the CD8 MHC TCR complex for the duration required to initiate T-cell activation. By contrast, the absence of receptor proximity in naive cells prevents CD8 from facilitating TCR-MHC complex formation, thereby impairing antigen recognition and diminishing cellular sensitivity. In conclusion, this non-invasive method operates under low excitation power and enables cross-correlation analysis of 2 to 4 proteins in living cells from tens of minutes to several hours, provided cellular conditions are properly maintained. It provides quantitative assessment of stoichiometry of non-constitutive complexes and their dynamics. At the same time, the method uses a simple sample preparation and user-friendly microscope operation. Altogether, it offers a robust platform for live cell molecular investigations and marks a significant step toward visualizing the previously inaccessible.